In pharmaceutical manufacturing, cleaning is not just about appearance. Cleaning validation guarantees that no residues remain, ensuring absolute patient safety and product quality.
This stainless steel surface is officially certified residue-free, yet remains coated in active ingredient streaks that only become visible under targeted fluorescence. The regulatory mandate of cleaning validation requires the absolute prevention of cross-contamination between different drug batches manufactured on the same equipment. Following Foreman and Mullen criteria, manufacturers observe two limits. Concentration must stay below 10 parts per million, and no more than 1/1000th of a maximum daily dose can carry over. These are supported by a qualitative requirement: the equipment must be visibly clean. Analysts measure these trace residues using high-sensitivity assays, most commonly total organic carbon, or TOC, and high-performance liquid chromatography. These laboratory instruments are blind to the physical state of the manufacturing floor. They can only detect the material that is successfully transferred into the sample vial.
The physical sampling interface creates a specific point of failure, where a compromised sample generates a false negative, despite laboratory precision. In one instance, a manufacturing site attempted to validate the cleaning of a polymeric matrix residue. The task was assigned to a newly trained technician who had not previously worked with this specific formulation. The technician followed the steps of the SOP, but did not account for the volatility of the organic extraction solvent used to dampen the swab. Because the technician moved too slowly across the large equipment surface, the solvent evaporated before it could dissolve the residue. This caused the API to rebond to the steel surface rather than transferring to the swab head, resulting in an analytical report of zero contamination for a vessel that was still dirty. A separate site encountered a similar failure when a safety mandate prohibited personnel from placing their heads inside a mixing tank. To comply, operators used a 12-foot TOS-scoping extension pole for sampling. The extension pole made it impossible to exert the precise, continuous pressure required for a valid swab, leading to inaccurate results that missed heavy residue deposits. Accurate recovery requires adherence to a specific sampling geometry for total surface contact. The procedure begins with an initial overlapping edge-to-edge pass. The operator then flips the swab, executing a perpendicular pass, and finally repeats with a second swab at 45-degree angles to capture remaining traces. Strict adherence to this physical geometry is a technical requirement for maximizing percent recovery. The metric that verifies the analytical results are representative of the equipment's actual state. Beyond the motion of the operator, the material science of the swab itself affects the assay. Standard cotton or nylon materials often introduce too much background interference for trace level detection. Total organic carbon assays are